do. All right. Hi, everybody. Let me get some stuff up. Looks like most people are here, which is good. Where is that gone? It's in there somewhere. Here we go. Come on. All right, so I'm going to crack on because I have to get out of here by 12, ideally, because I've got to go all the way over the other side of the city to get to an appointment by 1, which is an incredibly important appointment that I cannot miss or be late for, or anything, actually. So I'm going to just dive straight into it. Let's share my screen. I think that'll be easier because I'm going to bounce around a little bit here. So aim of the day today is to kind of field any questions about the format of the labs, like where stuff is, how you're going to be doing your assignments, you know, the partnering up and things like that, which some of you have already asked me questions about that already, but obviously some of you have not, because this is the first time we're meeting. We also need to go through the safety briefing briefly, and because you've probably seen this already in other courses here or elsewhere. And also go through the kind of signing and sending back of your safety acknowledgement form, right, which is really important, and I have to get through all of those and, you know, send them to the lab coordinator. So which one? All right, I'm just, come on, go away, go away. Think. Yeah, this is you lot. All right, I'm in something else for you lot anyway. So first things first, if you want to know when due dates are for different things, what you're going to be doing on a particular week, check out the schedule. The syllabus will tell you what you get for what different assignments, essentially, and, you know, a few other bits and pieces. Your student pairs is who you're going to be alternating with. So that's somewhat fixed in terms of who you're paired up with, simply because if I change the pairings, then I have to change the pairings of another pair as well, which is, it gets kind of awkward, like a follow-on effect. But I leave it up to you which week you want to attend. So if you really want to attend, like, you know, that Thursday isn't good for you, then feel free to swap with your partner. The ones that I sent out are really just, well, I had to pick one, right? You know, because otherwise it wouldn't make any sense. But if you want to swap them around, feel free, I don't mind. But once you do that, you need to stick to that, right? You can't just kind of have one student attending like 3 or 4 weeks in a row and then the other one, because that would just kind of mess things up a bit, to be honest. And it'll also mean that you don't get as much of the experience as we hope you would too. Anyway, so all of the due dates for the different assignments, or the due weeks, which is probably a better way of putting it, are in here. Generally speaking, the assignments that you have, and I'll go show you one just now, or show you where it is anyway. So this is where all your information is, and this is where all your assignments, are a couple, will be. And so generally speaking, your assignments for a given lab, and not all labs will have assignments, but most of them will, more or less, will be due by Thursday midnight the following week. Right, so I actually messed up a couple of these and I got that wrong, but I've since changed them. So the assignment for today will be due by midnight next Thursday, right? That also gives you a chance to ask any questions, particularly those that are not present at any particular lab because it's not their week. It will give you the opportunity to ask questions about that lab before that lab assignment is due. Right, so that's kind of like an extra opportunity if something doesn't make sense or you want to clarify something to get that fixed before the assignment comes due. I'm not going to go into this because if I do, it's going to show you all the answers. But this is where your assignment is for today. Essentially, it is almost a copy of some of the questions in your actual lab handout. Right, so this is your lab handout for today, and it looks something like, oh man, I closed it. What did I do that for? Oh, let's get that open up again. Come on. Doop, 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 doop. Here we go. And this would largely be the case for most of these assignments, by and large. So these would be normally what we would get you to print out and bring to lab. Well, actually, if you're attending lab, you need to print them out, obviously. But these are normally what you would fill in and return for your lab grade for that particular week. We can't do that because of a variety of reasons. And so you will be handing that in essentially virtually, right, through those assignments. And so this lab, not all of the questions on here are going to be in your assignment. If they're not, don't worry about them. They're there for your information. But for example, these questions are, right? So because we're not present in lab this week, I will give you these height vectors, right? So when you open your assignment, you can actually do that now if you wish. There's nothing to stop you. You'll actually see in the first question the height vector for the females, right? So there'll be 7 data points. That's called the vector. And then you'll be calculating mean, median, so on for that vector for those questions in your assignment. So essentially, the way that I've done it, and this was largely from doing this over the summer entirely online, which is a little bit weird, but you will be essentially doing the same as you would with your lab handouts, but you'll be doing it online in these assignments, right? So if you go to next week's lab, which a lucky half of you will be counting corn. It's so exciting. All right. This will be your assignment for lab next week. It's grayed out here. This is like professor view, basically, because you can't see it yet, right? So it will magically appear, I think, Sunday afternoon. So I actually set all these up on a Sunday afternoon, which is why the magical date is a Sunday afternoon. But every week, your assignment for that week will appear the Sunday prior, right? So you just look through those and it looks like there's nothing there and you might worry, but don't, because you will see them appear when they need to, right, for those individual labs. So this is where you will find everything. The only exception to, or exceptions to these online Blackboard assignments, I mean, there's another one in here, for example. Where is it? You know, this will be your assignment for the pipetting lab, let's say. The only exceptions to that are the assignments for the fly lab, right? So you'll be doing essentially a semester-long project with Drosophila. And the first half of the semester will be a cross experiment. So you'll be actually crossing different flies together to produce different offspring, and you'll be looking at linkage between two genes. And so all the information for that you need is here. So later on in the semester, I think it's one of the last things you'll be handing in, is a mini lab report for this project, right? And a little bit earlier than that, you'll be drawing a graph in Excel of your data to kind of illustrate your data, right? So those are the two kind of non-Blackboard assignments that you'll be emailing me directly with your name in the file. All of this stuff is done as individuals. Even though you'll be working with a partner to collect fly data or do the labs, all of the work that you submit must be your own. Obviously, you're going to be working with each other to do the analysis and talking about your lab reports and stuff. But if I see that your lab reports are the same, you don't get a grade for that section of your lab report, right? Because that's plagiarism. Right? So even though you will be working in partners, even if you might never actually meet your partner, which is kind of weird, you need to submit your work as your own, right? So anyway, if you want to have a little look about what I expect in your lab report, have a look at this. If you want to have a little look at what you'll be doing, check this out. We're not going to get started on that for a few weeks. And when we do, we will be going over these things in lab anyway, right? So these are really kind of supplements to what you'll get as in-lab instruction and on Zoom. They're not meant as a replacement. They're just, if you didn't get me telling it to you, then you can check these out and that might give you a different perspective. So that's really how it's going to work. It's going to be a little bit odd. I no doubt about that, because I've never done a half and half lab before. I've done entirely in person and entirely online, and I don't much favor the online version. It's a pretty weird thing to do. So hopefully this half and half is going to get you as much of the benefit as possible of doing in-person lab with as little of the risk as possible given current situation with COVID-19. So that's pretty much that. Does anybody have any questions about how the lab's going to roll, like the mechanics of it right now?
I have a question.
You'll need to speak up a little bit, love. I can't hear you very well.
I have a question about if we have like your lecture before lab and it's 15 minutes between, can we go to the library to watch your lecture before lab?
Yeah, you can do it wherever you like. I'll actually be on campus for that, which is kind of weird. So you'll see a different background because I obviously can't get from here to there in 15 minutes or even an hour. And some of you have already emailed to basically say that you might not be able to make the lecture in person because you'll be traveling in for the lab. That's also fine. The lectures will be posted to YouTube after, so you can catch up on anything you missed. So really, whatever works best for you, to be honest. Cool. Does anybody have any other questions about any of this stuff? If anything kind of occurs to you after the event, just send me an email and I will reply to you as soon as I'm able to. So let's get. Someone had a question?
Yeah, I had a question about pairing up with lab partners. Was there a list sent out? Who's going to be pairing up?
I can even show you where it is. It's just there. So this was the list sent out for student pairs for this lab. And as I said, it's up to you which week you attend. It's not a rigid list in that sense. But if at all possible, I'd like to avoid changing around the partners at this stage because it'll just be a fair amount of work and a little bit of confusion to sort that out. But if it is a very pressing issue, then we'll figure it out. You know, if you have like a blood feud or something with your lab partner, then I can sort that out. But hopefully that's not going to be the case. Good question from Destiny, if I've pronounced that right. So the idea here is that I will be doing this Zoom thing every week for the lab, for the first part of the lab where we talk about background, talk about the assignment, any work that needs to be handed in, any future plans. So if you are not attending that week in person, I very strongly recommend either attending the Zoom session or watching it afterwards. So you get all of the information about the lab. The only thing that you'll miss is not doing it, right? In the physical sense, right? You're not going to be there with a pipetta, kind of pipetting out liquids and stuff. Then after the lab, if any results were gathered during that lab by your lab partner who was present, be it a gel picture or fly data or anything like that, then you need to get that from your lab partner to be able to do the assignment, whatever that assignment may be for that lab that week. Also, this Zoom session will be posted to YouTube after the lab, so you'll be able to go back and check. You can, had a good question yesterday from my other lab section. You can even FaceTime or Zoom or Skype or whatever with your lab partner for particular aspects of it if you wish. I think that would be an awesome idea. And you'll be doing the exact same assignments. It's just one of you will be there physically, the other one will not, and that will swap every week. But you should have the same background information, you should have the same data, and you will have the same assignments. We're just varying who comes in to reduce the risk. So hopefully this way, at least you'll get at least some of the hands-on experience of doing this. Even if you're obviously not going to get all of it, you'll get some of the benefit, right? Which you wouldn't get if this was entirely online, which is what I did over the summer. So yeah, good question. But the work that you will submit, and this is worth pointing out because different people do hybrid labs differently, the work you submit every week will be the same whether you are there in person or not, right? So the same assignments, the same deadlines. If you have any questions about those assignments and you weren't in person, you weren't able to ask questions, you know, after the Zoom session finished, let's say, then you'll just email me or wait until you come to lab next week, and then you can ask me in person. I can go through anything for you. Right, so hopefully you're not going to be disadvantaged in any way by not physically being present. That's the aim. Okay, so let's get back to course content. Okay, so the other thing that I need to do today is quickly go through the safety briefing, particularly the bit about COVID-19 specific safety aspects, which will obviously be different this semester than prior semesters. And something I need you lot to do is to download this PDF, and you need to download it. Even though you kind of can fill it out online, it doesn't save properly for some reason. I don't know why. So download this to your computer or tablet or whatever, fill it out, sign it, save it with your last name and then course name, so I know who it is, and then upload it to this assignment, right? There's no grade associated with it, although I'll nag you incessantly until you do it, if you don't do it by Friday. But this will allow me to download all the safety forms and pass them on to our lab coordinator, who needs to keep a record of these for like legal reasons. Okay, so please do that as soon as you can. I'd recommend doing it straight after lab because then you won't forget and it'll be easier. But you do need to download it first and save it. Otherwise, you might have issues with, typically it hasn't been saving people's signatures. for whatever reason. You may also need to use Adobe Acrobat to fill it out and sign it, which is going to be unpleasant because I think Adobe Acrobat sucks, but you can just uninstall it afterwards if you need to. If you have any problems, you can actually just print it out, fill it out by hand, and then, take a picture of it and send me that, or upload that instead. But whatever you do, please do make sure that you put your last name and your course name in the file name. So save it that way. Makes it a lot easier to organize these things. Okay, so in terms of safety briefing, the main deal here is I'm not going to go through all of it because you've seen a bunch of it. There's actually some like fashion advice, which is rather nice. Don't those look good? But the main deal here is the efforts that we and you will go to really minimize the risk for catching COVID-19 by attending in person. So we have a bunch of plexiglass shields in the lab right now. They finished installing them, or most of them, last week, and they've got a few more coming in this week. And the aim is to physically, separate you off from other people sitting at the bench. We're only going to have 3 people at each bench that could seat 6, kind of seating diagonally so that you're, spaced apart. You also need to bring a lab coat with you. We are not going to be renting out lab coats for exactly the reasons of, potential contamination and stuff. So if you do not have your lab coat, you cannot attend class, right? Which would really suck. So next week isn't such a big deal because we'll be counting corn, and corn are not very dangerous in really any sense, unless you throw one at someone. So that should give you some time to get your lab coats. Students generally get them off Amazon. There's a variety on there. You can also get them at the bookstore. Don't think I've ever actually set foot in a bookstore, so I've never seen them myself. But other students have told me that you can get them there as well. So make sure you have your lab coats by week 3. They're an essential part of your safety equipment. Now, because of COVID, pardon me, this is a little bit specific to the GenBio 1 labs, but you need to bring your lab handout, print it out. I'm not going to provide them for you. Obviously a pen, lab coat. We have disposable masks for you, which you can grab when you come in. And the idea is that you then dispose of them afterwards, right? So that, to reduce the risk of transfer and contamination. You will not be allowed in the lab if you're not wearing a mask, right? So whatever your personal views on mask usage are, it is a requirement that you wear one in the lab for everyone else's safety. There's a whole bunch about how to wear masks, right? You don't put them over your eyes or your ears, put them out of your mouth and nose, and, obviously. I think we've all had plenty of practice at wearing masks by now, to be honest. You will also need to basically clean your work area. We will also be doing this, and we'll be cleaning any touch points, like centrifuges, stuff like that. But you also need to make sure that you do that yourself before and after use, like your personal lab space. We'll try and keep you distanced as best as possible. We're going to be spreading the equipment out over the lab so that there's less need to congregate in different places. We also have disposable gloves, nitrile gloves, which I don't think they're needed for any of the labs. Maybe the electrophoresis labs, it's generally a good idea to use them. But if you want to use those for your personal safety to dispose of when you leave the lab, you're welcome to do that too. All of the rest of the stuff, is similar to any other lab that you've taken. And it's generally common sense. So in terms of clothing, if you wear open toe, well, hang on, there, that's a better one, snazzy heels. Not entirely sure why these aren't appropriate, maybe because they're a bit too short. But if you're wearing open-toed shoes, shorts, I don't know, ripped jeans, which are kind of dubious from a fashion standpoint anyway, in my opinion. But if you're wearing any of this stuff, you will not be allowed into the lab, right? And this is really to protect you in case of an accident. Lab coat, obviously, if you have long hair or beards, then those need to be tied back or restrained. This is a new thing for me, so I'm not entirely sure how I'd manage it. I don't think it's long enough yet to worry about it kind of flapping into a Bunsen burner and the like, but we'll see how it goes. If there are any accidents of whatever sort, there's no consequence in terms of, you know, me being cross or anything, but you must tell me about them immediately and let me clear them up. So if you break a glass container, which is the usual kind of accident that happens in our labs, don't clean it up yourself. That's my responsibility. Because if I hurt myself, then, you know, it's kind of tough shit on me. But if you hurt yourself, you get hurt and I also get in trouble as well. So it's better if it's just me that does stuff. And the rest is fairly normal stuff. Oh, what's that meme about? I haven't seen that one before. Anyway, just ask. All right, that's the golden rule. Anything and everything, just ask a question, and that's it. So have a look through this. Fill out your safety form, upload it to Blackboard, and you're good on that front. Okay. Anybody have any questions about any of that so far? Good question, Lydia. So hopefully there won't be any issues with lab partner attendance. One thing that may come up is, you know, your lab partner's sick or they can't get childcare that day, or, you know, some chance occurrence. And if that's the case, you're welcome to attend in their stead, right? The main reason for splitting this up is to reduce lab numbers, right? So if you have enough notice and you're able to, which is hopefully the case, but it may not be, you're welcome to attend in their place. If that's not possible for whatever reason, then I'll make sure that you have data from another lab partner, pardon me, in order to be able to complete the assignment. Yeah, so you will not be disadvantaged if your lab partner is not able to attend lab for whatever reason. Yeah, so there's going to be wrinkles that we need to iron out as we go through this, because it's a new situation, but I'm pretty sure we can figure it out as we go along. All right, so oh, another question. Delaney, that is 100% correct. So whatever week you choose to attend in collaboration with your partner, the following week you will be attending online the Zoom session and not attending in person. Then the week after that, you attend in person, you do all the hard work. Week after that, you attend online. The week after that, you'll attend in person, and so on until the end of the semester. Yeah. As I said, it's going to be weird, but that's the best way we can do it. That we can do these semester-long labs and still manage the risk of infection as best as possible. Because if we didn't do it like that, if we did it like the conventional hybrid mode, then we wouldn't be able to do the big fly labs, because there's no way we could manage that so that everybody, you know, it worked. It would have meant completely remaking genetics lab for this semester, and I didn't think that was good for you, and it certainly wouldn't have been good for me. Okay, so moving swiftly on, right? Because I want you to get all of this stuff as best as possible, and I want to get to my appointment on time without hopefully any issues with crazy San Antonio traffic, which never ceases to amaze me. All right, so essentially what, where are we going? Get out of it. You can see my folder of cat and dog pics now. Get off. I don't want to see that. And we go back here and then go back here. There we go. Back to where we should be. So the issue with that we're going to start dealing with here, and we'll be doing this both overtly and kind of not accidentally, implicitly, I guess, throughout the whole semester. This is our very much an overlap on quantitative skills. And so really, as you transition to being biologists, right, which is really what you are or will become, you know, really becoming at least somewhat trained scientists, right? I'm not going to pretend that you're, you know, kind of ready to go out and get the Nobel Prize straight after undergraduate, but you'll certainly be on the path to becoming trained scientists. Part of that is to deal with data in a quantitative way, right? So it's all well and good saying, I saw this, right? Or this looks like this, you know, that kind of qualitative analysis. And right, that's often what initial discoveries come from, right? You look at a Petri dish and you go, that looks weird. Ta-da, we have ampicillin, right? First antibiotic. Or, you know, any other kind of observation, right, which looks interesting. And I'm, you know, I do plenty of those myself. But if you want to convince anyone else that that observation is true and applicable to other circumstances, or other scientists will be able to repeat your work if they attempted to do so, you need to be able to analyze those data quantitatively, right? And so one great example that I saw recently was a job candidate talked about how he saw two monkeys at two different cameras in a forest in Colombia. And that was it. So, well, that's interesting. Great. What does it mean? Can you reproduce it? What's the data, right? How do you back that up? You know, if you want to really convince me that that's something fundamental, you need to do a bit more work than that. So that's one reason behind this need to be able to demonstrate basic quantitative skills and to use them, right? The other reason is that biology and science in general is noisy, right? Especially biology. Actually, mathematics, theoretical mathematics, no noise whatsoever. It's pure. Biology is quite the opposite of that, right? It's not pure whatsoever. There's a lot of stuff going on. And in my opinion, it's more interesting as a result. But the challenge that kind of gives us is kind of sorting the wheat from the chaff, right? Being able to truly identify something interesting going on and to be able to convince others of that, right? So this really is the first step on that path, essentially. And another, just to show, kind of showing off a little bit, I guess. But another thing is that for a lot of the data that you collect, you're going to collect reams of data, right? Reams being like stacks of paper if it were in paper format. You know, so how do you condense that and not simplify it, but simplify it in a sense, right? How do you break that down to its simplest components so that it's easy to understand, right? And so this is a great example, in my opinion, partly because it's work that I've done, but partly because there's a whole bunch of numbers on this page, and we've got multiple pages. right? So this is a gene expression study that I did this summer for 10 different genes, which were differentially expressed in different mutant backgrounds, right? You got the mutant backgrounds up here. And so essentially I had a company carry out quantitative PCR on these genes to figure out if those data that I collected before were correct, right? And so these are all the different genes, right? And these are the expression ratios of the gene relative to controls and stuff like that. And I looked at these when I got them, I was like, whoa, that's interesting. Yeah, there's definitely something going on here. And then my eyes started bleeding and like, I was like, ugh. And I basically stopped, because what I need to do is I need to go back to these and actually start generating means, calculating standard deviations, graphing these data, so I can end up with a nice clean graph and go, this is what's happening, right? For this gene, but not this gene, and for this genetic background, but not this genetic background, and so on and so forth, right? And so if I don't do that, then these data are essentially useless, right? If I try to present data in this format to a scientific journal, they would just laugh at me. They'd be like, no, go away. Put this in a format that our readers can actually access, right? And so this isn't just kind of like an abstract concept that we're talking about in this lab. It's really what scientists do every day, or at least I should do it every day. Sometimes I just don't get time. But that's not through a lack of desire, but more just coping with different things in my life, teaching wise, work wise, that is. So really what we're going to be starting doing here is start talking about the different ways in which you can do that, and there are various benefits and pitfalls, right? And so essentially one of the ways in which we take those data, right, repeats, let's say, or multiple sample points for a population or a subset of the population that you have collected, and we condense that down into a mean, right? And so if you have, and the data for these questions are in the Lab 1 assignment, which is on Blackboard. You'll need to figure out what the mean is, right? So what's the average height of the people that I've given you data for, right? The problem is that once you take all of those data, right, those collection of data points, and you condense it down into a single number, into the mean, you lose all the information about that population. And you might think, well, why does that matter, right? It matters because you can have very different populations have exactly the same mean, right? Or at least samples, right? So this is a little bit of a tricky thing. When I talk about population, really what I mean is like the large group of which you're going to be sampling a subset of that, right? So whenever you collect your data, your sample, pardon me, we're going to assume that you have normal distribution for that sample, right? Because otherwise all kinds of funky stuff happens. But even if you have normal distribution, this kind of classic bell curve, you can have populations with the same mean, but very different population makeups, right? So for this example, this black line, this sample here, what you can see is that all of the data points in this sample cluster quite closely around the mean, right? So there's very little variance. So an example would be, I don't know, you calculated or sampled the heights of a, is it NBA? I'm not very good with US sport acronyms, basketball team, right? Most likely they'll all be really tall, right? And all tall, right? Not like, you don't have like one real short person in a basketball team and mostly tall people. They're all tall people, right? So there'll be very little deviation around the mean for that particular population. but that's not necessarily going to be same as the larger population, right? Where you would expect a much kind of more spread out distribution of individuals around that particular mean. But as soon as you break that down into a mean and calculate that, you lose all that information, right? And so if you just compared means for the height of those different populations, you would miss an awful lot of information, right? And so one of the things that we want to do is to start figuring out what's the variation around the mean, right? And that's really one of the things that we'll be doing today. And so at first instance, that's the variance, right? Which is a somewhat straightforward calculation. It's basically the difference between the mean and each sample point squared and divided by the sample size -1, right? Which is what we've got down here, right? So mean is really easy to calculate, and I'm sure you've all done that before. Essentially, it's just all of the data points added together divided by sample size. And median is the middle number, right? So let's see, what have I got? So I have this all done from yesterday. So if this is our data set here, right? So we've got 5 different heights, sample size n of 5, then the mean is simply all of those added together divided by 5. If you want to use, and I think I've uploaded this. No, I haven't. Let me just double check. I have not. I will do that in a moment, either right after this or when I get back from my interview. But here we go. What was I thinking of? Just lost my track of thought for a moment. So you can do all of these questions by hand if you wish, or you can use Excel. Mean is easy. Median you can do also by hand. You simply take these data and you sort them in order, right? So this is your vector. A vector is simply the collection of data, right, from large to small. You line that up from large to small, and your median is your middle point. Or you can be either efficient or lazy, depending on your point of view, and you can use Excel to do that. This is what I do, because I view myself as being efficient. So if you want to calculate the mean, all you do, and as I'll walk you through this as well, quickly, make sure we have enough time. Hit equals. So whenever you hit equals in a cell in Excel, and again, I'll upload this Excel spreadsheet in a little bit, so you can use this to calculate with the actual vectors, right, which I've provided you. As soon as you do that, well, why didn't it do it this time? Well, I don't know. That's kind of weird. Anyway, you should have something pop up in this box, right? And so these are all of the different functions you can use. So where's average? They call it average, not mean for some reason. Now, what will happen now is it will make its own best guess as to what you actually want, which is sometimes right, but often it isn't. So you can either change this manually by clicking here and then just changing the cell number, right? So we want cells B1 to B5. Or you can go to this box. Hopefully you can all see, yeah, you should all be able to see what I'm pointing at. Click on this, and then you're able to double click and drag. Oh, did I do that wrong? I did, didn't I? Let's get rid of that. Right, this is what it should look like when you do it from scratch. Double click and drag, and you will select the box that you want to calculate that from. And as you can see here, that gives you the same thing. Hit return, and then return again, or enter, and that will get you your mean. Median, you can also do the same, selecting that as a function, is fairly straightforward. Now, variance, as is shown here, this is the equation for variance, right? And this may look a little intimidating. It's not meant to be. But really what this is this bit here is saying sample point minus the mean squared. So we have here 5 sample points. So we're going to do 5 calculations of sample, which is going to be 187, minus the mean squared, right? So you can do that by hand, or you can do it here, and this is actually a clunky way of doing it. This is a better way of doing it. I just remembered this morning while I was eating my breakfast. That should give you the same thing. It's just a lot easier to write. I don't want to do that. Thank you very much. So the up arrow 2 is to the power of 2, so it's squared. Should actually get you exactly the same thing. It just looks neater. Pardon me. So what I'm doing right now is I'm actually calculating all of these x, which is sample, whatever that sample is, minus the mean squared, right? So that's what I'm doing right now, which you can see up here. What I'm doing is I'm just doing it in a neater way than I did before. I don't do that very often, so I often forget those functions. Other things I do a lot more frequently, so I'm more familiar with them. Right, but
sir?
Say again?
Sorry to interrupt you, but
No, no worries.
what about the part where you have to take that?
Take what?
The variance, that is. You know, like how it's, like it
Yeah.
says, the difference of the sample minus the average, and then square that. There's also you have to divide it by n-1.
Yeah, exactly. The thing is, it's very hard to do that in a Excel formula without confusing yourself. At least I could probably think about it if you gave me like 20 minutes and a cup of coffee. But I'm essentially doing this in steps, right? So these are all of the individual variances squared. Now what you need to do is you have to add those together, right? That's going to give you your sum of the variance, right? Which is this. So you can just do equals sum and select those like so. Or you could do this by hand with a calculator, also works just fine. That's going to be the top half of the equation, but that's not the sample variance. To do that, you need to do exactly what you said, which is the next one. This is this calculation, which is that sum divided by the sample size, which is 5 minus 1, right? And so if you wanted to do that in Excel, that's how you would write that formula. So one day I'll actually get around to figuring out how to do this as an actual, I mean, I probably could, but I don't have time. So I don't want to kind of go off on a tangent because I'm trying to keep to time here. But you could quite easily figure out an equation to do all that in one step. I've simply done that in a couple of steps to kind of work through it for you. So one of the issues with calculating variance or sample variance is that issue of squaring the difference, right? So you have to square the difference because by its very nature, half of your data points, more or less, will be less than the mean. So when you have a smaller number minus a large number, you'll get a negative result, right? And you can't have negative centimeters because you'll be like into the ground. So we have to square that to get rid of the negativeness or the negativity. But when that carries over to your units. So when you calculate sample variance, you actually end up being an area rather than a height, which is, clearly nuts. So we need to fix that, right? And so you won't find really ever anyone talking about sample variance in a description of their data for this exact reason, because it will create funky units that don't make any sense. So the actual unit, or the measure, sorry, which is a better description used, is standard deviation, right? And that is, it's a real simple formula. It's simply the sample variance expressed as a square root, or it is the square root of the sample variance. So you see this part here underneath that line, or underneath that big square root symbol, is exactly the same as this one up here, right? So all you do is you take your variance and you square root it, right? So there you go. You could do that as a function. So squirt is square root in Excel speak, and all you do is equal sqrt, and then you point at the variance, the sample variance. Oh, fudge nuts, I've screwed it up. I have to put stuff in brackets, otherwise it gets all kind of confused. There you go. So that's one way of doing it. To be completely honest, I never do it that way. What I do is I just work it out as standard deviation first. The reason why I'm taking it through this is because I want you to understand what standard deviation actually means, right? It's the square root of the sum of the differences squared divided by sample size minus 1 there, which is a very long way of saying it's how stuff varies, right? So you can actually calculate standard deviation straight up, right, which is what I always do. And that simply equals STDEV, right, which is Excel speak for standard deviation. And then you select, oh, what did I just do? Not what I wanted to do. The same data set that you've been using all along. Hit enter, and that gives you the exact same result. So whichever way you do it, you should get exactly the same result, either doing it in Excel, doing it in by hand, right, which is perfectly valid. We don't have many data points in this vector, so it is feasible to do. As you can imagine, I wouldn't want to do that for my data set because it would just, I'd be an old and haggard man by the time I'm finished, and I don't want to be an old and haggard man. Or you could do it by Excel via one of these two routes, right? You do need to work out the variance because that is one of the questions that you will be asked, right? So you can't completely get away with using Excel. Where is it? Variance, variance, variance. Here you go. So one of the questions that you'll need to ask in your lab assignment will be, what is the sample variance for those two vectors? Again, because we're not in person this week, every time I ask you a question, you'll need to, I give you the vectors, right? So you don't have to write them down or whatever. But my hope is that you actually stick those into Excel and use Excel because, you know, it's a lot more efficient way of doing it. Okay, so essentially though, standard deviation is a simple measure of variance, but it doesn't really take into account sample size. I mean, it does use sample size in the calculation, but it doesn't really necessarily account for how big your sample is. And the big deal here is that whenever we do an experiment, whether it's a molecular biology experiment like the one I showed you, or you're going out into the hill country and counting lizards or whatever, right? Whatever aspect of biology you happen to be partaking in, it's almost a certainty that you'll be unable to measure the whole population. It may be because that population is too large. It may be because they're hiding under a rock and you can't find them. But whatever you do, you will very rarely, if never, measure the whole population. And so really, what you're doing is making an argument that your particular sample set is representative of the larger population. That's part of what we're doing here. And inherently, I shouldn't need to tell you this because you should inherently know it yourself, but it's worth stating explicitly, is that the larger a fraction or percentage of the population that you sample, the more confident that you are that your mean is actually representative, right? I mean, that makes sense. If you measure the height of 2 people in your class, you're not going to be as confident that the mean is the same as the whole class unless you measure half of it, let's say. And you can take that to whatever length you wish in terms of sampling. And so one way of adding a degree of a measure of confidence, not a degree of confidence, a measure of confidence, is to calculate the standard error of the mean. And really, what that does is that gives you a reward almost, right? It's not actually intended as such, because, you know, we don't reward people for doing good science. It's just, well, we do. We give them prizes and stuff. But in terms of confidence, your confidence gets bigger the larger your sample size. And the way we do that is that we take our standard deviation and we divide it by the square root of the sample size, or the square root, which is, let's go up here. Right, so this is the standard error of the mean. It's simply the standard deviation, or the S, divided by the square root of the mean. And so what I've done is I've calculated the standard error of the mean for this data set. Remember, this is not the data set that you'll be using, so you can't just copy these figures over. It's not going to work. But I've calculated the standard error of the mean, or SEM, for this data set, which is standard deviation divided by the square root of 5, which is 2.24, right? And just for illustrative purposes, right, not because this is what I'm asking you to do, but I also calculated the SEM for, let's just as a thought exercise, if that population consisted of 20 sample points, not population, that's sample rather, right? And so now you're going to be dividing the same standard deviation, one presumes, or similar, right? But now you divide it by the square root of 20, which is 4.47, right? So your standard error of the mean gets smaller. And so essentially what you're saying is, rather than your mean for the population, well, it's going to exist somewhere between these two extremes, right? +5, or 180 plus 5 centimeters and minus 5 centimeters. So what you're saying is that your population, the bigger population, the mean is going to be between 175 and 185 centimeters, right? That's essentially what you're saying with those statistics. Now, if you had a larger sample size of, say, 20, now you can be more confident that your mean, rather than being within these two limits, now it's going to be between, I don't know, 175 and 182 and a half, let's say 183 centimeters, right? If you measure the height of 100 people, right, now your SEM, or standard deviation, will be divided by 10. Now it gets even smaller, right? So essentially, the closer you approximate the larger population, the more confident you are that the mean in that population is within those limits. Which also means that if you were to do the experiment again, but with a different 100 people, then you would expect the mean for that sample group to also fit within that smaller range, right? So that's essentially what those different metrics, I guess, functions are talking about, right? So I'll post this up to Blackboard. I'll probably do it right after I go, so I don't get confused or forget. But if this is all just gibberish to you, that's fine. I can answer questions, or you could just do it by hand, right? Which also works. For those kinds of sample sizes that we're working with, that's totally fine, right? So whatever works best for you. I would simply suggest getting familiar with Excel, because it's a very powerful program to use. Yeah, Priscilla, good question. This assignment is due midnight next Thursday, which is, I think the 3rd. Let me check. Yeah, that's the 3rd. So midnight of the 3rd is when this assignment is due. Okay, so the very last thing that you need to do for this lab, and I'm going to be a little quick, is, oh, you're welcome. Numbers are hard to visualize, right? We're visual creatures. We interact with things visually in a lot of ways. And so this is not the last step. This is an important process because we're essentially taking these data down and we're generating our assessment of these data, right? In terms of mean, we're describing it, right? But what you need to do next is you need to draw this out. You need to make it visual. And if you only have two vectors, male and female, fine. But imagine if you're looking at different age groups in that population, or different races, or different socioeconomic backgrounds. Now you're going to start having multiple figures, and trying to visualize those as numbers is really tricky. So the last thing you need to do, ta-da, trusty Rusty, here's my black pen. So the last thing you need to do is draw a graph, right? So you can either draw it in Excel and upload that to the assignment, or you can draw on paper and you can take a picture and upload that. Whatever works best for you, I really don't mind which, as long as it is legible, right? And so I personally have advised against using crayons because those are really difficult to write. I know, I'm just finishing up, love. Right, so you want to draw a graph, 2 lines, super simple. You're going to have males and females, or females and males, doesn't really matter which. You'll have your y-axis, which is going to be height, units, centimeters. And that's probably going to be, I don't know, 160, 170, 180, 190. I don't know for sure because I can't remember what numbers I gave you, but it's probably around that kind of range. So you need to plot the means. So let's say like so. I'm making an assumption here. I can't remember what data I gave you. Could be that the females are higher. Doesn't really matter. Right? These are the means because those are, the mean is, you know, 178 for males, let's say. Then you need to show your confidence range. So you need to plot your standard error of the mean, right? So that is always plus whatever the SEM is, let's say 5, and minus 5 for the males. Females are going to be, maybe they have a different SEM, maybe they don't, but you will also plot that here as accordingly. Right, so you need to do this for the vectors that I gave you. You're going to calculate the means, the SEMs. You need to draw the plus and minus SEM, and you need to take either a picture of that and upload it, or upload the Excel spreadsheet where you did that. And that is your assignment for this week's lab. These questions and the graph. All right, I've got to run, I'm afraid. So I'll be back at my computer in a couple of hours. So if you have any questions about any of this, please do email me. If I'm not able to answer those, then hold them for next week's lab and I'll answer them then instead. But now I have to go get in something smarter. Data for height are in the assignment questions themselves. As soon as you open it up, you'll see it. All right. Awesome. Sorry for the rush. Again, if there's anything that doesn't make sense, please do ask me questions and I'll get back to you as soon as I can. All right. Have fun.